All right, so for my presentation, I'm going to be talking about phenocopying and specifically phenocopying potential in vertebrate gastrulation development. So I'll talk more about that and what that kind of means as the presentation goes on. To start, however, we're going to start with some keywords. First keyword is, what is a phenocopy? The way that I kind of like to, identify what phenocopying is when the phenotype of one organism that develops in an unnatural external environment mimics or copies the phenotype of a different genetically different organism that develops in a normal environment. The term gastrulation basically is an essential process in development of all vertebrates in which a ball of cells known as a gastrula forms three specific cell tissue layers: the ectoderm, mesoderm, and endoderm. This is a very crucial part of development and it allows our cells to be more specialized and can differentiate further into the different components of our body. The third keyword I like to describe is what is a primitive streak. And a primitive streak is a collection of cells that forms a vertical line of mesoendoderm tissue in the central midline of the gastrula. So if we look at the top picture up here, we see the gastrula cross section, and that kind of like folding in on itself is like kind of like a primitive streak. It's kind of like the midline part of mesoendodermal tissue. In the picture in the below, we see that yellow line represents that is a primitive streak, and any further yellow lines will be primitive streaks in this presentation. A couple more keywords. One, which is intercalation. Intercalation is basically the ability of cells to exchange places with their neighbors. When we have gastrulation development occurring, there's a lot of cell movement. Cells are intercalating with each other, moving between neighbors and stuff like that. Ingression, on the other hand, is another type of cell intercalation within gastrulation development, where epithelial cells specifically will migrate away from the epithelial cell they're with and become mesenchymal cells, basically kind of being able to differentiate into other forms of cells. And we're going to talk about perturbations, which is basically an effect on an outside influence of a system from its natural occurring path. So imagine you're going to work and you take the same path every day, and that is the normal path that you're on. If a train goes in front of you during your path, well, that is a perturbation. It is an outside influence on a system. Your system is getting to work, and a train basically disrupts that natural system that occurs. Next, I'm going to talk about kind of the summary of the results and the conclusion quickly, just to kind of outline what the main part of this study is. The study that I looked at was called "A Mechanochemical Model Recapitulates Distinct Vertebrate Gastrulation Modes." In other words, the researchers in this study developed a mechanochemical model that uses lots of physics and very intense formulas, which I'm not going to get into. But this model, or simulation rather, used in the study can successfully predict the outcome of perturbations in gastrulation development of a chick, and can also be used to mimic the gastrulation of vertebrates by slightly changing the parameters and variables of this model. So on the right here we see four different species of animals. We have reptilians, we have amphibians, we have fish, and we have a chicken. And basically, to sum it all up, this model that they used, you can change the parameters and the variables of this model to recapitulate and mimic the different gastrulations that occur in these different species, all primarily from looking at a chick gastrulation. I'll get into more of this. Outline for the rest of this presentation, we'll quickly talk about the background of the presentation of the research, talk about the hypothesis, what the testers are trying to, determine, what they hypothesize. Talk about the results, the conclusion, and we'll finish with bibliography. First off, I'd just like to do a quick background. This is a little recap from the previous presentation of Ben's. The researchers in the first part of this paper talk about the dynamics of gastrulation in the chick is a result of myosin activity that drives this tissue movement. So the cells during gastrulation of a chick embryo move in a very specific way, and it's a very predictive way. And they determine that, they believe that the results of this movement is driven by myosin activity. Myosin is one of the very important molecules that's in our muscles. It allows us to contract our muscles. And so it's that same kind of process which moves these cells as they develop and as they kind of move around, and I guess during the process of gastrulation. So using a planar chemical model which they have discovered, or I guess which they have developed, the dynamics of chick gastrulation and the development of the primitive streak is predictable. So these cells move in a very predictable manner, and they're able to map those cells out and trace it over again and again and create this physics formula that basically create a simulation that can show the gastrulation. So in these images here, we see on the lower part is an experimental look at the development of a gastrulation of a primitive streak here with the P and the A. The left side is kind of prior to that, different types of cells, the blue ones being different type of cell than the yellow ones. And we can see above that the model, that the type of mechanochemical model or simulation that they made is able to mimic like the natural development of natural gastrulation of a chick fairly accurately. It looks very, very similar. So where we can take this model and how we can look at different ways that the cells, the gastrulation of chick embryos, basically the perturbation of different chick embryos, how we can mimic that with this model. And that brings me to my next slide, which is the hypothesis. So basically, the hypothesis in this study shows that varying the initial conditions and parameters of this model, or the simulation, allows for the phase space of gastrulation morphologies to be seen across vertebrae to be recapitulated, which is consistent with other experimental observations of gastrulation development in other species. So in other words, we were able to study the effects of these perturbations or deviations of the natural path in experimental trials, and then take the model that was developed and mimic it, or I guess change the conditions of the model, the parameters and all of the fine numbers and stuff like that, and basically mimic the very similar development on the model, similar to what happened experimentally. So the study observed 4 different perturbations in chick development, resulting in the formation of 4 different primitive streaks, or I guess primitive streak structures. The first perturbation we talk about is twin development. So this actually occurred like naturally and spontaneously. Sometimes twins would form, and twins happen kind of naturally in our environment as well. And basically what would happen is a mesoendoderm precursor area would split into 2, resulting in the formation of 2 primitive streaks. So this is an experimental video, experimental trial of the chick embryo that went through twin development, and we can see here that there are 2 primitive streaks that develop. Now, if we look at the twin development through the model, this was consistent, this idea of 2 primitive streaks developing with the mechanochemical model. Once they were able to basically change the parameters, we can mimic what looks exactly like 2 primitive streaks. As you can see here, 2 primitive streaks developing in a very, very similar fashion as the experimental one. We talked about, or I guess in the perturbation number 2, they used axitinib. Axitinib is an inhibitor for vascular epithelial growth factor receptor, which is a big part of many signaling pathways. And when axitinib was induced, it prevents active cell ingression, resulting in the development of a shorter and thicker primitive streak. So in an experimental trial with the gastrulation of a chick, the development here, we can see the formation of a shorter and thicker PS when in the presence of axitinib. Now, this is very similar, again, with the mechanochemical model. The effects of this primitive streak development could also be predicted using the model. So we change the parameters of the model, and again, we can see the development of a shorter and thicker primitive streak. And so basically the whole point of this paper is that we are able to mimic when perturbations occur in a natural streak development, and we've created this mechanochemical model, we can still change the parameters and mimic those perturbations, those kind of deviations off the natural path it's a very predictive way. The third perturbation we're going to talk about is FGF2, which is the addition of fibroblast growth factor 2 in the experimental test results in the development of a mesodermal ring that kind of acts like a circular primitive streak. So in this video we see basically the development of a circular primitive streak. Again, when fibroblast growth factor 2 is introduced, it kind of causes a different formation, a perturbation, a deviation off the main type. And if we look at the model, again, changing the parameters of the model, we can basically look at, we can mimic basically the formation of a primitive streak, a circular one, and how that, with the cell movement and stuff like that, the model can be used to also mimic this circular primitive streak. Talk about perturbation number 4, which is LDN and CHIR. LDN is an inhibitor for bone morphogenic protein, and CHIR is an inhibitor for glycogen synthase kinase 3, both used to induce the formation of, again, another ring mesoderm. However, this time, this treatment was able to block cellular contraction and cellular ingression, or apical contraction, and cellular ingression in the primitive streak, but had minimal effect on the intercalation within the entire development, causing a collapse in tissue at the end. So in this experimental video, we see kind of like the quick formation of a mesodermal ring, but then because the inhibitor, or these inhibitors didn't allow intercalation to occur, had a minimal effect on it, the tissue would collapse. And here again on perturbation number 4, we have LDN and CHIR. With the model, we can again, the mechanical model can also predict this, formation of a circular mesoderm, and it also predicts the cellular collapse. In the left-hand corner here, we see the mesoderm kind of produced, and on the right-hand corner up here in the graph, we see how this model can kind of show this collapsing of tissue as time goes on. So this mechanical model can be manipulated in many different ways, changing its parameters and all that, and can mimic all of these different things. So where does this all go in the end? We connect it back to phenocopying as being the idea that an organism in a different environment can develop in a way similar to another organism in a normal environment. So what's kind of cool is that this mechanochemical model that they've created can mimic gastrulation in other vertebrates by altering the parameters of this model. So the primitive streak structures can resemble the grooves observed in reptilian gastrulation, as we can see here, of that thicker and shorter primitive streak. The circular streak structures can resemble the germ band of teleost fish gastrulation, and even the collapsing tissue resembles the movement occurring during the closing of a blastopore in amphibians. So these are all the development of gastrulation from other studies that the researchers have looked at. But basically, the idea that this model that they have created, this simulation, can be changed and can mimic different gastrulation patterns in other vertebrates. So what does this mean? This means that basically this research suggests that relatively small changes to the environment in which development occurs and gastrulation occurs can cause, changes in cell behavior that are linked to development across vertebrates. To say that in another way, there are environmental factors that play a role in development And when we change those environmental factors on, specifically chick gastrulation in this case, we can see how that mimics basically the development of different vertebrates in their own gastrulation in a normal type. And so this is really cool because it brings us one step closer to understanding how phenocopy mechanisms work. When we have environmental alterations, how those change on a different environment can change and mimic. And basically, it'll help us understand how potential there are different pathways for organisms, and those different pathways can maybe allow us to connect the different evolutionary pathways that have occurred in the past and stuff like that. So this study is really cool because it kind of brings us one step closer to understanding those phenocopy mechanisms. And quickly, these are the sources that I used. Thank you very much.